Hello, everyone, and welcome to today's live broadcast. Does your protein analysis toolbox need a refresh? Presented by Rich Jones, PhD. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Beckman Coulter Life Sciences. Beckman Coulter develops, manufactures, and markets products that simplify, automate, and innovate complex biomedical testing. More than 275,000 Beckman Coulter systems operate in both diagnostics and life sciences laboratories on 7 continents. For over 75 years, our products have been making a difference in people's lives by improving the productivity of medical professionals and scientists, supplying critical information for improving patient health, and delivering trusted solutions for research and discovery. To learn more about Beckman Coulter Life Sciences, please visit www.beckman.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Rich Jones. Dr. Jones has a long-standing interest in the development and application of automated protein analysis technologies and workflows for interrogating protein systems in human biology and pharmacology. He has more than 20 years of experience in the field of protein and cell biotechnology. As an assistant professor in the MedMay Department for Cancer Research at the University of Chicago, Dr. Jones developed micro-Western arrays for studying the responses of cancer cells to therapeutics and for understanding chemotherapeutic resistance mechanisms. He was also a core fellow and founding member of the Institute for Genomics and Systems Biology and founder and director of the University of Chicago Micro-Western Array Protein Analysis Core Facility. Dr. Jones's complete bio is found on the LabRoots website. Please join me in welcoming Dr. Jones. I will now turn the presentation over to him.
Thank you very much, Judy. It's a pleasure to be here, and I welcome everyone to my presentation entitled, Does Your Protein Analysis Toolbox Need a Refresh? So in today's talk, I will be providing a survey of protein analysis tools. I won't be diving too deeply into each of them, but depending upon the feedback that we get, I may present more deep analysis of specific areas of protein analysis tools in the future. So the presentation that I will describe today is a bit of a meshing of graduate level courses that I put together while at the University of Chicago and while a teaching fellow at Harvard University over the years. And again, I welcome your feedback so that we might go in more detail in the future to help you all in your own work. So in today's presentation, I will provide an overview of protein analysis methods, and I will use the life of the biotherapeutic as a framework with which to introduce and discuss these methods. And so for the purposes of today's talk, we're going to follow the life of a prototypical biotherapeutic from the stage at which discovery is occurring, biological discovery in cells and testing hypotheses, onward to the course of expression and purification of proteins, to further doing binding analysis and surveying protein interactions, and then onward to biophysical characterization. We won't talk so much about downstream work in manufacturing. So firstly, why are proteins important? This is near and dear to my heart. It's what I've spent most of my life studying. Proteins represent the machines encoded by our genomes, and by all genomes that perform nearly all of the functions of living systems. They range from quite small sizes for insulin and other types of growth factors, but they can also be quite big, as an example of the collagen fibers, where proteins assemble into larger, higher molecular weight complexes. And again, what is quite important is that they link our genomes with our health. And so when things go wrong with protein systems, it's typically a fundamental root cause of diseases. So I describe here the epidermal growth factor system. It's responsible for many types of cancer, including breast cancer, which the HER2 therapeutic is a target, and non-small cell lung carcinoma, for which the EGF receptor is a target. And in this particular system, a growth factor, the epidermal growth factor, binds to the epidermal growth factor receptor. In many cases, it leads to the heterodimerization with other partners like ERB2, which is also known as HER2, the breast cancer target. This leads to activation of receptors on cell surface, and it tells them to grow. There are a number of Sark homology 2 so-called proteins that recognize the modifications on tyrosines subsequently, and it leads to a lot of events downstream that ultimately cause the cells to grow and metabolize quicker. And in many cases, when we get older, these systems go awry, and it leads to cancer. So proteins can undergo extensive modification. If you just start with our genome that has about 20,000 genes in it that can be alternatively transcribed and spliced into different mRNAs, which can then be translated alternatively and modified alternatively, and it leads to high complexity of proteins, and it requires a variety of complex tools to understand the systems when they're working, and then also to try to understand how they go wrong. So here's an artistic rendition of the platelet-derived growth factor receptor in red binding to the yellow highlighted platelet-derived growth factor receptor on the cell surface, and you can see little antibodies that look like little V-shaped molecules. And this is an artistic rendition of the complexity of types of molecules that we're studying and trying to understand for the development of therapeutics. So as I mentioned, aberrations in protein signaling networks lead to diseases. On the left, I depict a cell signaling network that I studied while I was an assistant professor at the University of Chicago, and it was subsequently published in Cancer Prevention Research, where we studied the action of honeybee hive propolis, which is material that bees use to stitch up their hives. And we used microwestern arrays to understand the networks that were impacted by the addition of honeybee hive propolis or feeding to mice. And we studied their impacts on tumor cells. And when the tumor cells were growing, they would have activation of a series of signaling networks that led to their growing more quickly and detaching from cells. And when we applied honeybee hive propolis, it more or less turned off their ability to sense insulin, so it shut down the insulin signaling network. So in therapeutics, biotherapeutics, where we're developing molecules, often we're trying to develop neutralizing antibodies to the ligands that are attaching to growth factor receptors. Sometimes we're trying to prevent the dimerization of the receptors themselves or their activation, or their small molecules that are developed which inhibit the kinase domains of these growth factor receptors on their intracellular surface. Sometimes there are inhibitors designed to prevent the heterodimerization partners such as HER2 or HER3 with the epidermal growth factor receptors. So as I've approached the study of of protein systems, I show here a graph that describes the understanding of individual proteins, and this is typically performed with biochemistry and biophysics methods, where individual proteins are expressed, purified, and subjected to a variety of tests to understand in quite detail the individual molecular events that lead to protein activation. At the other end of the spectrum, we have proteomic tools to understand whole proteomes. In the middle, the area that I've spent a lot of work developing tools, I call proteinomics, and this is helpful for understanding of biological processes where we want to understand lots of proteins, typically on the order of 100, in order to focus our efforts on understanding biological processes as best we can without diving too deeply into individual proteins. So this brings up an important point, which is the scale at which you are conducting your research. And it's important to, when you're going into trying to solve a biological problem, to understand at what scale. On the left, I show individual pipettes and then multi-channel pipettes, and as we get farther to the right, you start to have the need for automation to do multi-channel pipettes and multi-channel liquid handling in order not only to speed up the process, but more importantly to reduce the variability associated with the results. Because as you begin collecting hundreds of data points or thousands of data points, it's quite critical to reduce the variability so that you can be able to compare data across time and across individual experiments. So here I provide an overview of the life of a prototype biotherapeutic, and it's this overview that will serve as the foundation for the talk today. We start off with target and pathway discovery and the tools associated therewith, and next, the protein expression, purification, optimization phase. Then I'll discuss the screening of proteins for binding targets or modulators of protein targets. And finally, I will discuss a series of methods associated with the more deep biophysical characterization of proteins. And I won't spend so much time talking about, or I won't spend any time, in fact, talking about the process scale-up or the downstream processing. So at the beginning of the life of the biotherapeutic, we have the target pathway discovery phase, where we're trying to understand the biology that exists in normal biological processes and that is dysregulated during the course of disease. Many of these methods range from immunohistochemistry that's often used in the clinic to bead-based analysis methods, cell lysate arrays, Western blots, a Coulter principal method for studying cell sizes, capillary and microfluidic electrophoresis for understanding size and charge, flow cytometry for studying proteins in cells, and mass spectrometry for studying protein abundance and modification. So digging more deeply, immunohistochemistry allows one to examine the relative expression and location of protein in fixed tissue sections. It's important in the clinical analysis of tissue specimens for diagnosing disease states, the phases of cancer. It tells you the protein's relative amount and its location and gives you an indication of cell and tissue morphology associated with diseases or pathology in tissues. But it doesn't tell you charge, shape, molecular weight, protein purity, folded state, isoform, the polydispersity, or also known as the size range of the proteins. And so these other pieces of information will have to be gleaned with other protein analysis methods. This is a quite old method, originally described in 1941. The next phase we have newer methods, including bead-based methods in which many proteins can be examined simultaneously. Because these beads are barcoded with dyes that have a specific ratio of dyes, these ratios of dyes allow one to know the identity of the protein under examination. And these beads are labeled with antibodies that are able to capture the proteins of interest, and then secondary fluorescent antibodies are used to quantify the abundance of bead or of proteins that are attached to the beads. So in theory, 100 or so proteins can be examined simultaneously through incubation of specific beads with cell lysates. In flow cytometry, one is able to examine proteins in cells or on cells, again using fluorescently labeled antibodies that bind to specific protein targets. Lysate arrays, Western blots, micro-Western arrays, and capillary microfluidic electrophoresis are different variations on the theme of applying proteins to different substrates and then examining their abundance in either the context of glass slides or in gels. So the first method I'll talk about here, the cell lysate microarrays, were described in the early 2000s and subsequently evolved a bit in the mid-2000s. And here I'm showing work from Saveka et al. That they described in Nature Methods. And in this method, you take lysates from cells, and you can do this with hundreds of cells treated with small molecules or therapeutics or growth factors, and array those lysates onto a variety of slides that can then individually be interrogated with antibodies to different pan-specific, meaning measuring the total abundance of proteins, or with phosphospecific or other types of modification-specific antibodies. We subsequently further evolved this method in 2010 and described a manner in which protein or microarray methods were used to array the lysates onto acrylamide gels that were then subjected to semi-dry electrophoresis and then transferred to nitrocellulose and probed in 96-well format with 96 different antibodies. And then we showed that the signal that we got from these arrays was linear with regard to abundance of proteins, and that they were analogous to macro-Western blots. But the cool thing with micro-Western arrays is that you could use a lot less protein than you would normally use with a Western blot, and you could perform these experiments in a more automated manner in microtiter format. So in this study, we just compared the response of growth factors of cells, A431 cells stimulated with epidermal growth factor at following either 0 minutes or 60 minutes, and we probed for beta-actin to measure just total protein content in the green channel. And then we followed the modification state of a series of tyrosines on the epidermal growth factor receptor itself or downstream targets of the epidermal growth factor, and then monitored the fold changes in the phosphorylation at different time points with either large macro-Western, traditional Western blots, or micro-Western arrays. And this is what this looks like in a 96-well microtiter format. And I show here the phosphorylation kinetics of S6 ribosomal protein at the top left, which undergoes very slow increases in modification after growth factor stimulation, versus the epidermal growth factor receptor itself below, which shows very rapid phosphorylation kinetics within a minute of stimulation. And then also we probed all of the wells with beta-actin simply to show that we had equally arrayed proteins on our slides. And we asked in this study what the network-wide response of cells was to stimulation by epidermal growth factor. How do the cancer cells respond globally to changes also in nutrition? If we simply added more glucose or amino acids to cells, cancer cells, how would their signaling networks respond? And how also would they respond to difference in growth factor signaling networks, or different growth factor signaling levels? So this is a very busy slide, and I apologize for that, but we were quite proud at the time at the ability to track so many modification states of important signaling network proteins. And that's what I show on the right of the chart, is all of the different phosphorylation states that we were following at the same time. And the important thing to note here is simply that if you look at the very top of the chart, we show 0 nanograms per mil of EGF added, and then we monitored at 0, 1, 5, 15, 30, and 60 minutes. This is simply adding new media to cells. And when you look at this, you see very few membrane-level proteins at the top that are changing over the course of 1 minute. And in fact, many of the changes you see that are depicted in white hot at the bottom are proteins associated with insulin, AKT. These are proteins in the cell that very slowly get upregulated when the cell senses a change in sugar or amino acid content. If you look at the left part of the chart, as we go from 2 nanograms to 50 to 100 to 200, you start to see very intense activation of the growth factor receptors on the cell surface within 1 minute. And then they decay, but then they come back at 60 minutes. And so this is, if you look in simply the 1 minute panel at the top, those were proteins that underwent maximal stimulation within 1 minute. Then in the next series at 5 minutes, these are another series of the MAP kinases that are stimulated downstream of the growth factor receptor itself, and that are responsible for initiating cascades associated with cell growth. So in this study, in summary, we quantified 92 phosphosites of different signaling proteins associated with cancer and cell growth. We probed with different growth factor levels at 6 time points. We collected about 24,000 data points, and this was the most data points ever collected with Western blots at the time of the study in 2010. And this is simply a zoom-in of the things at 1 minute that were maximally stimulated, the receptor tyrosine kinases themselves, non-receptor tyrosine kinases at the membrane, as well as tyrosine phosphatases and adapter proteins that turn off the signal. So I'll quickly move on through this slide. Ultimately, we took the data and wanted to build a bit more of a higher level network. And so we worked with researchers at MIT, the Laufenberger Laboratory, to use mathematical inference methods to take our data and build a causal inference network that provided insight into the connectivity of the proteins in the pathway, and could subsequently be used to try to understand which of the proteins might be most important in shutting off the signal, such as you might want to determine if you were designing a therapeutic and you wanted to shut off or find the most sensitive part of the network. So when you're collecting protein modification data, you many times also want to collect cell phenotypic data that you can use to relate the responses of the proteins in the cell to actual physiological changes in cell size. So I show here the multisizer 4E. It uses the so-called Coulter principle, where cells go through an aperture, and the change in resistance as the cell passes through the aperture is used to count the cell, and it's also used to determine the sizes of cells. And this is often used in the context of cell therapy to monitor the changes in cell size as T cells are stimulated to undergo maturation. They'll change from about 10 microns to 30 microns, and in many different physiological contexts, the cell volume will change, and the Coulter principle, such as is used in the multisizer, can be used to follow that. When one wants to study proteins on much larger scales, in terms of thousands of proteins in modification states, mass spectrometry is often used, and this is used in the context of more proteomic studies. And through LCMS, liquid chromatography mass spectrometry, proteins are typically separated based on hydrophobic interactions, such as with a C18 resin. All other methods for chromatography are gel filtration, where proteins are separated by size, ion exchange, where proteins are separated based on their charge, or affinity, such as protein A, where a specific protein is immobilized on a column surface and then used to specifically pull out proteins based on affinity. So getting back to mass spectrometry, typically a protein is digested and the peptide mixture is put into a mass spectrometer, and the charge-to-weight ratio is examined and then referenced against a database in order to examine or identify abundances of proteins or changes in abundances of proteins and modification states in the context of cellular biology. So after a specific target has been identified, then typically a workflow moves forward to express either the protein targets themselves for further analysis or to make antibody-based biotherapeutics, which I show here in the yellow. And typically a cell line is developed and the health is monitored with something like the ViCell that will, through Trypan Blue, measure percent viability during the cell culture process, or the Metaflex, which measures the changes in the glucose and other metabolites. Usually there's some centrifugation step depending on the process size, but at small scales, typically a centrifuge is used to pellet cells, and then there's some process of purification via column chromatography and following or tracking the expression with acrylamide gels and or the functionality using enzyme-linked immunosorbent assay or ELISAs, and then some subsequent follow-up with mass spectrometry to confirm the size and the glycosylation state of the therapeutic proteins. So let's talk a bit more about that. So as I mentioned in the beginning phases, in many cases you may pull out several clones of different antibodies or proteins that have been modified in different ways to try to optimize the cell growth conditions. And the ViCell from Beckman Coulter is a way that one can, in relatively high throughput, monitor the viability of cells in an automated way via Trypan Blue staining. Again, the Metaflex will measure glucose and several other metabolites that will give you an indication of the metabolic state and the health of the cells during the process and help you to optimize particular conditions that will give maximal protein expression. So cells are typically pelleted via low-speed centrifugation and then clarified, or the supernatants clarified with high-speed centrifugation. And then often in the early stages, antibodies, for example, are purified rapidly via protein A affinity chromatography, and then maybe subsequently cleaned up via ion exchange chromatography and then maybe finally purified via gel permeation, or also known as size exclusion chromatography. And often during the process, the ability of each of these steps to enrich the purity is often tracked either by polyacrylamide gels, where the proteins are separated by size, and then examined via either Coamassi staining or subsequently perhaps by silver or colloidal Coamassi to give a more sensitive readout. Western blots are also used to confirm that the particular proteins being examined are the protein that you are actually trying to follow. And so you would typically generate antibodies specific to the protein of interest for the purposes of immunoblotting. Functional assays such as ELISA, also known as enzyme-linked immunosorbent assay, which was developed and first described actually simultaneously in different publications by Perlman and Engvall back before I was born, a long time ago. In this method, one either attaches to a microtiter well an antigen of interest if you are expressing an antibody and you're looking for function, and the antibody can either be labeled with horseradish peroxidase or a fluor that will allow you to track the functionality of the antibody by its binding and its colorimetric readout on the slide. And there are different ways to perform this, either directly, where an antigen is bound to a slide and then read out by binding to an antibody that's labeled, or indirectly, where a secondary antibody is actually labeled and then incubated subsequently with the initial antibody that you're screening. Or alternatively, the sandwich ELISA is performed, in which one antibody is coded on a slide, then an antigen is bound. And typically with this format, you're actually measuring the functionality of an antigen, and you would take plates that are coated with a capture antibody for your antigen, and then you would subsequently add more antibodies to the antigen of interest, and via this sandwich format you can get much higher selectivity for the functional antigen that you are expressing. So LC-MS is often used to confirm protein size integrity, modification state, and also the glycosylation of proteins. And there are different flavors of mass spectrometry depending upon the situation. Often matrix-assisted laser desorption ionization, also called MALDI, is used in cases where one wants to simply characterize very quickly the integrity, the size integrity of a peptide or protein. In this case, it's immobilized onto a surface, and I show here in the middle bottom a peptide that's been put onto the surface of a gold slide and then hit with a laser. And typically these peptides will pick up one positive charge under the conditions, and then one simply times the amount. This is all automatically, but the machine times the amount of time that it takes to accelerate your peptide until it hits a wall, a photomultiplier tube. And through this process, one then calculates a size to charge ratio, which I depict in the spectra on the top. Electrospray, also called ESI, quadrupole mass spectrometry, is usually integrated in line with some sort of liquid chromatography. And I depict the process in the bottom right, where a HPLC is spraying its contents into it through a nebulizer and into a quadrupole mass spectrometer. And in this case, the charge state of the proteins or peptides is related to the number of positively charged amino acids, such as arginine and lysine, and it's usually conducted under very low pH conditions so that all of the carboxylates are at that point neutral. And then one can, especially if you have multiple quadrupoles connected in tandem, you can fragment the peptides along the amide backbone and then read out the amino acid content of the peptides, like I show in the ESI quadrupole spectra at the top right, where the distance between each of the peaks is typically a function of the peptides that are fragmented along the backbone. And in an automated way, the instruments will read out the spectra and compare the spectra to spectra in a database in order to confirm the identity of the peptides that you are working with, or glycosylation patterns as well. So in phase 3, I've talked a bit about the panning for binders. So in an earlier publication as a postdoc, I wished to uncover the connectivity of proteins and signaling networks. And through this exercise, we cloned and expressed hundreds of sarcomology 2 domains. These are the proteins that recognize activated growth factor receptors. At the time that I started the postdoc, much of the genome had still not yet been sequenced very well, and many of the proteins in the genome still, the function wasn't clear. So we cloned all of the so-called sarcomology 2 domains, which have a certain motif that is predicted. It was predicted at the time to recognize phosphotyrosine motifs of growth factor receptors, and we wanted to query the interaction potential of all of the epidermal growth factor receptors with all of these signaling proteins downstream that we knew were most likely responsible for the cancer signal of epidermal growth factor receptor. So in this method, we use protein microarrays. In this method, we took the collections of the hundreds of SH2 domains and we arrayed them onto the surfaces of glass slides. And using this method, you're able to screen the interaction potential of hundreds of proteins that are on the slides simultaneously with hundreds of other fluorescently labeled proteins or peptides. Okay, my screen, okay, here we go. So in this context, we actually combined the previous technology of microscope slide arrays with the microtiter plate technology that's often used to automate through liquid handling robots. And what I'm showing here on the right is this merging of protein microarrays with microtiter technology, where each of the microarrays is contained within the well of a microtiter plate so that an automated pipetting robot can then be used to add different peptides for screening purposes. And what I am depicting here is a small amount of fluorescent dye that has been doped into each of the proteins that we arrayed onto the slide. And when we incubated our slide with phosphopeptides that were derived from the growth factor receptor, then we could start to see that there were different proteins on the slide that bound in different affinities to the peptide that was incubated. And when we used a saturation binding curve, we could fit affinities. There were some peptides that maximized their signal at very low concentration, such as the ABL2 kinase that bound to this particular protein of HER2, also known as ERB2, and the specific phosphorylation site here is tyrosine 1139. But we could also characterize, if we incubated at very high concentrations of the peptide, we would see saturation with lower affinity proteins, such as the breast cancer anti-estrogen resistance 3 protein. We had the midpoint of this binding curve was 1 micromolar versus 71 nanomolar for the ABL2 kinase. But because this was all done in an automated manner in terms of the microfluidics, or sorry, not the microfluidics, but in terms of the liquid handling and in terms of the data extraction via the image analysis, we are able to quickly characterize the binding potentials of all 150 of the putative phosphotyrosine binding proteins in the human genome. And we used these affinity maps to characterize for each growth factor receptor, the epidermal growth factor receptor, also known as ERB1 in the top left, ERB2 in the top right, which is today typically known as HER2. ERB3 at the bottom left really recruited the phosphoinositide kinases really well. Those were all depicted in red. And then ERB4, which wasn't well understood at all, and I believe today it's still a little bit not well characterized, but it's thought to play a role in the liver. This is characterized at the bottom right. So in panning for binders, arrays can be used. Also, the enzyme-linked immunosorbent assay that I described before can be used to pan for binders. It's simply with the ELISA approach, one typically is characterizing 96 interactions on a slide, versus with the microarray method, one can characterize thousands of interactions on a slide using thousands of fold less reagents. So finally, I will talk about biophysical and biochemical characterization methods. It's oftentimes, as I showed in these previous few slides, when you're performing any particular study, you will lean quite heavily on one particular method or one particular instrument. And it's very important in order to be a very well-rounded biochemist, biophysicist, to have at least some grounding in a variety of methods, so that when one sees a particular problem, one can quickly identify a series of potential solutions in order to move forward in your research. So in the first method I'll talk about for biophysical characterization is light scattering. So the Delcimax Core is an example of an instrument that Beckman Coulter sells, which uses both dynamic and static light scattering to quickly characterize the molecular weight and the size distribution and get an indication of the aggregation state of proteins in the solution phase without any label. And with light scattering, the correlation of scatter in time will provide an indication of the sizes of the proteins in solution. So I show at the top right an example of a large protein, or it could be a protein, or it could be other types of molecules. But the idea is, when light is scattering in time, it is quite correlated if the larger the protein is, because it's moving relatively slowly. As the protein, if it is smaller, it will actually be moving much quicker by Brownian motion, and the change in scattering patterns in time will change much quicker. And using the algorithms of the instrument, it will, in the matter of seconds, with a very small amount of material, as few as a few microliters, one can quickly obtain the molecular weight and the size distribution and the polydispersity, or the range of sizes of proteins in solution, and quickly gain an understanding of the quality of protein that you're working with. The Delcimax Pro is an example of an instrument that uses both light scattering as well as a voltage potential application in order to derive a charge from proteins. at the point at which formulations are developed, often the charge of the proteins in solution is critical in order for determining whether or not the proteins will be stable in solution because of their repulsion for each other, or whether or not they will tend to coalesce and agglomerate and fall out of solution. In many types of industries, for example, in water quality, they're trying to find conditions in which the particles are neutral so that they will coalesce and come out of solution during the water purification process. So with a particular Delsimex Pro instrument, it has a couple of interesting modules that are very important in the context of proteins. The first is the so-called assist module. So typically, as you're applying a potential to a solution that has salt, there will be bubbles generated due to the electric current that will generate bubbles. And because bubbles scatter light, they interfere with your ability to collect zeta potential measurements. And with the assist, it actually pressurizes the flow cell so that the bubbles don't form, and therefore you can actually measure the zeta potential or the charge state of the particles in cases where you typically can't. The other interesting feature of the Delsimex Pro is that it has 32 detectors at different angles which collect scattering information in parallel. Because proteins are very sensitive, if with a standard zeta potential measuring instrument that's not been designed for proteins, you will typically require several minutes in order to collect enough light scattering data under the conditions of alternating electric current before you can gather enough information with which to derive a zeta potential. Whereas with the Delsimex Pro, it collects the light scattering information in parallel for many detectors simultaneously, allowing you to collect the information within a few seconds and therefore be able to collect the information before the protein is destroyed or degraded due to the application of the electric potential. So during the biophysical and biochemical characterization process, surface plasmon resonance is also often used in order to measure in a label-free manner the binding of and the affinity actually of an immobilized protein on a gold surface with proteins that are flowing in solution. And so the first protein is typically immobilized, and then a second protein, small molecule, peptide is flown through a microfluidic chamber. And as more mass binds to the surface, it creates a change in signal that can be measured by the detector. And what one is typically then examining is a change from a base level response on the surface to a maximal level of response as a function of concentration of the analyte that's in solution. And through this method, one can determine on rates, off rates, and steady state binding profiles. More recently, surface acoustic wave technology has been used to generate slightly more information than the surface plasmon resonance method. And through this method, if you look at the binding profiles that I show in the top right, what you see are curves that look very similar to SPR. However, with the surface acoustic wave technology, using both the phase of the acoustic wave on the surface as well as the amplitude, one can examine not only changes in binding, but also changes in the conformation of the protein on the cell surface. And again, you can perform kinetic analysis, and you can perform this in a label-free manner. Microscale thermophoresis is yet another way to measure protein interaction potentials or small molecules, peptides. This does use fluorescence, so but there are two different flavors that this can be performed. In one manner, intrinsic tryptophan fluorescence can be used to read out protein fluorescence, and the other, you can attach a fluor to the protein of interest. But in any event, with this method, a laser is used to create a temperature gradient, and depending upon the binding state of your protein, the larger it is, the slower that it will migrate along the temperature gradient. And therefore, if one performs a titration series from low protein to high protein with a constant amount of the fluorescently labeled version, one can then quantify a saturation binding curve, as I show in the bottom right. Whereas ligand concentration goes up, the speed at which the protein is migrating out of the thermophoretic gradient goes down. So another flavor of this interaction analysis is biolayer interferometry. With this method, the proteins are actually immobilized on the surface of a tip, and a interference pattern along the tip is measured as proteins are binding to the tip. And so this tip is typically dipped into 96 or 384 well plates, and the interference pattern along the tip is then examined. And if you look at the sensorgram at the bottom right, you'll see a graph that looks very similar to SPR and the other methods that I've shown. You'll go from a base state where nothing is bound to the tip. As you begin to dip into solutions that end up binding to the surface of your tip with its immobilized proteins, you'll start to see an increase in the intensity of the signal in a relationship to the affinity of your analyte for your proteins. This is, again, label-free, and because it can be done in 96 or 384 well plates, it's quite high throughput. Another important tool is multi-angle light scattering. So with multi-angle light scattering, this is typically performed downstream of size exclusion chromatography as a way to get an additional corroboration of the size of a protein. So typically the spectrograph or spectrochromatogram, rather, that one takes off of the size exclusion chromatography is overlaid with a series of sizing measurements taken during the online run. And in many cases, the protein shape will make it perform aberrantly on the size exclusion chromatogram. Some small proteins will elute and they will look larger, and vice versa. And the multi-angle light scattering downstream will allow one to have a second added level of confidence that the size readings obtained from the chromatogram is consistent. And in cases where they're not consistent, one can dig deeper to try to understand what the actual sizes of the proteins are. Circular dichroism is another method that one can use to understand the folded states of the proteins and to compare batches of proteins. If you look in the top right, you see spectrum of the green and blue protein from different batches of proteins that are slightly different. And similarly, at the bottom right, you see a natural versus a recombinantly expressed version of protein, and they have different spectra, and it's difficult to know exactly what the difference is, but it ultimately is a different reflection of the ability of the protein to absorb left and right polarized light, and the alpha helices, the beta sheets, etc., given their asymmetry, will absorb right and left-handed light in different manner. And if you compare batches of proteins in time, you will be able to see that they're either the same or different depending on their ability to rotate the light using circular dichroism. Thermal shift assay is a method that one can use to determine the stability of proteins in different conditions. And so using a light cycler or a real-time PCR instrument with fluorescently labeled proteins, one can apply a temperature ramp and then measure the fluorescence under different conditions. So I show at the top right proteins that are melted under two different conditions that give slightly different midpoint melting temperatures. In the bottom right, I show the protein by itself or protein with a ligand and no protein at all, and you can see that the ligand actually causes the protein to be more stable, and therefore it takes a higher temperature before you see the change in fluorescence. So this is a useful for probing formulation conditions and conditions for which your protein may be more stable. So with isothermal titration calorimetry, one can very rigorously examine many different biophysical characteristics of proteins. So typically one will incubate a protein with another peptide or protein and then measure the amount of heat that is generated with small injections of the peptide or small molecule. And as the peptide is injected, heat will be released, and this heat in the sample cell will be compared to a reference cell, and then ultimately with the amount of heat absorbed, one can obtain many biophysical parameters associated with the interactions of peptides and small molecules with your protein of interest. So finally, I show analytical ultracentrifugation. This tool is extremely rigorous. It requires a bit more training than some of the other methods, but it's able to answer many questions associated with the shape of the protein by itself or with complexes, how the protein behaves in solution, its purity, its aggregation state, the association with other proteins, diameter, mass, stoichiometry, etc. It's an extremely rigorous method, and the Optima AUC is an offering from Beckman Coulter that provides this. In summary, proteins represent important therapeutic targets. There are a variety of tools. I've just talked about some of the more commonly used methods today. In the future, we may dive a bit deeper, and I welcome your feedback on some of the methods that you think would be more helpful to you in your biological research. Obviously, periodically refreshing your protein analysis toolbox and your understanding of each of these methods is critical for you to be able to see and identify which tool is most applicable to solving your problem of interest and moving onward and accelerating the pace of your discovery. And with that, I thank you, and I welcome any questions. Thank you very much.
Thank you, Dr. Jones, for that informative presentation. So it's time for Q&A. If you have a question you'd like to ask Dr. Jones, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. And while we're waiting for questions to come in, I would like to once again thank Dr. Jones for his presentation. Do you have any final comments for us today?
I welcome any questions you have about either today or in the future about workflows you have here at Beckman Coulter. We are constantly reaching out to folks in the biological community to try to understand and have a pulse on the solutions that you need to solve your tools in biomedical discovery. So I welcome those questions.
Thank you. Okay, well, thank you once again, and I would also like to thank our sponsor, Beckman Coulter Life Sciences, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.
Bye.